All right, students. So I figured I would show you a video of how to do this pre-lab that we need to do before we do the actual lab. So I'm going to share my screen here with you. So in this lab, so before we do that, one thing that I want to clear up are the parts of a cell that are involved in transcription and translation. So these are just important things for us to have. So on page 15 of your notebook, you're going to put these images. You're going to tape them or glue them, and we're going to use this information here to help us. So we know from before, if you looked at your old notebook from last year, we have two types of cells primarily. So we have eukaryotic cells, and we have prokaryotic cells. So eukaryotic cells, these are the ones that we have. So we humans have eukaryotic cells, so do plants, so do animals. They have eukaryotic cells. Prokaryotic cells, these are bacteria. Bacteria are prokaryotic cells. So what are the main differences between them? So eukaryotic cells, they're called eukaryotes because they have a nucleus. So eukaryotic cells have a nucleus. You'll notice here that prokaryotic cells do not. So this one does have a nucleus, and this one has no nucleus. No nucleus. Very sad. Okay. So what else? So we know that both of them have cytoplasm. So cytoplasm is that liquid that fills up the both of the cells. So they have cytoplasm. And what else do we have that's in common? Both of them have ribosomes. So ribosomes, right? This is where translation happens, right? In the cytoplasm, specifically in the ribosome. So both of them also have ribosomes. Okay. So if we look back at our notebook on page 11 and 12, right? We know that inside of the cell, we have a nucleus, and in the nucleus is where our DNA is. So our DNA is right there. It's in the nucleus for our cells, at least as humans. Bacteria, however, do not have a nucleus, so their DNA is just it's in the cytoplasm. But here, so right inside the nucleus, right, we have our DNA. Right, in this case, we have it in the cytoplasm. This here is DNA. So DNA, I kind of lied to you. DNA, the way that we had looked at DNA in our class was seeing it as a short molecule that only had like a few nucleotides, but DNA is actually a very long molecule. It has over 60 billion nucleotides. So our body likes to store things efficiently. It doesn't like, right, if you have something that's 60 billion nucleotides, that's really long. So what your body does is to store things more efficiently, it wraps up the DNA very tightly in a structure called a chromosome. So this here is a chromosome. So when someone talks about your chromosomes, they're talking about your DNA. We're just talking about, right, in humans, we have 46 chromosomes, so your DNA is split up into 46 different chromosomes. Why? That's just the way it is. Some people have one chromosome more, one chromosome less. There are some variations, but typically we have 46 chromosomes. All right, so here we're going to see just a piece of DNA. We're going to zoom in on one piece of DNA. One piece of DNA we call a gene. So this here is a gene. So when you hear gene or your genes, right, your genes have to do with sections of your DNA that code for a specific protein. Inside of these genes, right, we have two strands, right? DNA has two strands. These are both strands. And then there are base pairs, and those base pairs are formed with four letters, T, A, C, and G. These are called nucleotides. And if you have a pair of letters, so like C and G, for example, this here is called a base pair. So that's more just to say, right, so we're going from big to small. So if we think about these things from the smallest to the biggest, if we were to put these like on a scale from small to big, right? So the biggest thing would be a cell. Cell is the biggest. Then if we're right inside the cell, we go to the nucleus, and then goes the nucleus. And if we go inside the nucleus, right, we have our DNA. Our DNA is inside there. We have DNA. As we know, our DNA, right, our DNA is wrapped up into smaller structures called chromosomes. So chromosomes are smaller than DNA, even though they're the same thing, chromosomes. Right, one piece of DNA, just one piece of DNA that gives us some information. This part here, right, this is just one piece of DNA. This is called a gene. So then comes a gene. Right? Genes are made of these nucleotides. So then comes a base pair, right? A base pair is the union of an A and a T, right? A and T, or C and G. These are called base pairs. And then the smallest is a nucleotide. Nucleotides are tiny. Okay, so this is just to show you how this is going to work out. So the next experiment that we're going to do, so this, by the way, is on page 15 of your notebook. And this next one is going to be on page 16. So what we're going to do in this next lab is this. So let me add in this image. What we know, what we have learned before when we were looking, when we talked about antibiotic resistance in bacteria, is that the way that bacteria communicate with each other is through plasmids. So there's these little circular pieces of DNA called plasmids like this. That bacteria can share with each other, like they share DNA with each other in that circular form. And that allows them to communicate with each other. What they're actually doing is they're sharing genes that code for specific proteins. So this here is a plasmid, right? It's a piece of DNA or gene. Okay. So what we're going to do is right, what we know from transcription and translation, again, we're going to look back on page 11 and 12 of our notebook. Right, during transcription, we know we start with DNA, and at the end, we form a strand of mRNA. Oh gosh, it's a little hard to see. We form a strand of, let's put blur on this camera. We form a strand, oops. We form a strand of mRNA. So then in translation, that same mRNA comes to the ribosome and it becomes a protein. Right, first we form some amino acids and forms a polypeptide. So a group of amino acids are called polypeptides or peptide. And then we form a protein. So what we're going to do here is we're going to put that exactly right. So here we have, right? So here we have a plasmid. We're going to introduce a plasmid to bacteria. We're going to put, right, during transcription inside the bacteria, the plasmid or DNA is going to be converted into an mRNA. Then during translation, right, the mRNA, we find our start codon and we go through each of the codons. Then there's a tRNA that has an anticodon and it carries also an amino acid. So these are called these here. Each of these circles is an amino acid. A chain of amino acids we call a peptide. So this whole thing here, this is a peptide or polypeptide, same thing. And then when it folds, it forms a protein. So what we're going to do in this lab is we're going to give bacteria a plasmid or a gene for a specific protein, right? That gene, right? That DNA, as we know, DNA during transcription, this part here is called transcription, where we go from DNA to a peptide, or from DNA to mRNA, and then translation, right? That mRNA goes into the cytoplasm, or I guess in bacteria, it's in the cytoplasm already. That mRNA becomes a chain of amino acids or a peptide, and at the end, it becomes a protein. So what we're going to do is we're going to give this plasmid for a protein that's going to glow green. This is called green fluorescent protein, or GFP. It's just a protein that glows green. So what we're going to do, right? We're going to have our bacteria. This is my drawing of bacteria. We're going to have this plasmid that has the gene that has the information for how to make this GFP. And we're going to put it inside the bacteria so that the bacteria makes many, many copies or makes many, many green proteins. And then if it works, right, if this process works, if transcription and translation work, we should see the bacteria glow green. In case you were wondering, so mRNA, this M stands for messenger. So what happens is, right, mRNA carries the message from DNA on how to make a specific protein. So if there's a specific protein you want to make, you're going to find those directions in the DNA, and you're going to convert it to an mRNA, and then that mRNA will tell the ribosome how to make that specific protein. So in this case, we're doing basically the same thing. Let's make sure that carries the message from DNA on how to make a specific protein. right? In this case, we are going to be giving it a gene. Oops. We're going to give the bacteria a gene or a piece of DNA, a plasmid. In this case, we're going to give it a gene that's going to create a green, that is going to eventually create a green fluorescent protein. So we're going to go first through mRNA, and then it's going to be GFP. But that's what's going to happen in the next lab. So this should be on page 15 and 16 of your notebooks. And this is just to give you a view of what's coming next. All right. I'll see you soon.